My topic is High Performance Liquid Chromatography HPLC, presented by Mrs. Afroz Pathan, Assistant Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences. Coming to introduction, HPLC stands for High Performance Liquid Chromatography, sometimes referred to as High Pressure Liquid Chromatography. High performance liquid chromatography is a powerful tool in analysis. It yields high performance and high speed compared to traditional column chromatography because of forcefully pumped mobile phase. HPLC is a chromatographic technique that can separate a mixture of compounds. It is used in biochemistry and analytical chemistry to quantify and purify individual mixture of compounds. Coming to some definitions. Chromatography. Chromatography, it is a physical method in which separation of components takes place between two phases, a stationary solid liquid combination. Mobile phase. Solvent which carries the analyte. It may be a liquid or a gas. HPLC is a type of liquid chromatography where the sample is forced through a column that is packed with stationary phase composed of irregularly or spherically shaped particles or a porous monolithic layer or a porous membrane by liquid mobile at a high pressure. Next, principle of HPLC. To understand the principle of HPLC, we must first look at the principle behind liquid chromatography. Liquid chromatography is a separation technique that involves the placement or injection of small volume of liquid sample into a tube packed with porous particles, that is stationary phase, where the individual components of sample are transported along the packed tube by a liquid which is moved through gravity. The main principle of separation is adsorption, where the mixture of components are introduced into column of various chemical and physical interactions takes place between the stationary phase and the sample molecules, which leads to the separation. They travel according to their chemical affinities towards the stationary phase, and the component which have more affinity towards the adsorbent travel slower. The component which has affinity towards the stationary phase travels faster. Since no two components have the same affinity towards the stationary phase, the components are easily separated. HPLC is a separation technique that involves the injection of small volume of liquid sample into a tube packed with tiny particles, that is 3 to 5 micrometer in diameter, called as stationary phase, where the individual components of sample are moved down the packed column with liquid mobile phase forced to the column by high pressure, which is delivered by a pump. These components are separated from one another by column packing that involves various physical or chemical interactions between their molecules and the packaging particles. These separated components are detected at the exit of this tube by flow through device, that is detector, that measures their amount. The output from the detector is called as liquid chromatography, except the speed, efficiency, sensitivity, and ease of operation of HPLC is vastly superior than LC. Types of HPLC based on mode of separation: normal phase chromatography, reverse phase chromatography. So these are the two types based on mode of separation. HPLC working is differing. In normal phase chromatography, stationary phase is polar, that is hydrophilic, and mobile phase is non-polar. Whereas in reverse phase chromatography, stationary phase is non-polar, that is hydrophobic, and mobile phase is polar. Polar polar bonds and non-polar non-polar bonds have more affinity than polar non-polar. Based on elution technique, HPLC is divided into two types: isocratic elution and gradient elution. A separation in which mobile phase composition remains constant throughout the procedure is termed as isocratic elution. In isocratic elution, peak width increases with retention time linearly with the number of theoretical plates. This leads to the disadvantage that late eluting peaks will get very flat and broadened peaks. Best for sample preparations. often used in quality control applications that support or in close proximity to the manufacturing. Whereas in gradient elution, a separation in which mobile phase composition is changed during the separation process is called as gradient elution. Gradient elution decreases the retention of later eluting components so that they elute faster, giving narrower peaks. This also improves the peak shape and peak height. Best for analysis of complex samples, often used in method development for unknown mixtures, and linear gradients are most popularly used by using this technique. Based upon scale of operation, HPLC is divided into analytical HPLC and preparative HPLC. No recovery of individual component substance can be done in analytical HPLC, whereas individual components of substance can be easily recovered by preparative HPLC. Based upon type of analysis, HPLC is divided into qualitative analysis and quantitative analysis. In qualitative analysis, a substance in order to ascertain its nature of its chemical components can be done. We can separate individual components but cannot assess the quantity in this analysis. Whereas in quantitative analysis, we can determine the amount and proportion of its chemical constituents. Quantity and impurity of the individual components can also be assessed. This is the general flow diagram of HPLC, where you can see two solvent reservoirs, also called as mobile phase reservoirs, are first degassed so that we can remove any dissolved gases from the mobile phase. Then they are mixed and through a high pressure pump they are ejected towards the pre-column, which helps to remove unnecessary impurities which are already present in the mobile phase. Later on, it is sent to the main analytical column. In between, a sample injection port is kept where the sample can be injected. Based upon the interactions of the sample and the stationary phase, the components can be separated. So the components which have higher affinity towards the stationary phase will elute later, and the components which has less affinity will elute first, which can be detected in the differential detector. And whatever the excess mobile phase and sample will be sent to waste, and an LC, that is liquid chromatogram, will be developed by which retention time of the separated components can be detected. Coming to the parts of HPLC. First one is solvent delivery system, also called as mobile phase. The mobile phase in HPLC refers to the solvent being continuously applied to the column or stationary phase. The mobile phase acts as a carrier to the sample solution. A sample solution is injected into the mobile phase of an assay through an injector port. As the sample solution flows through the column with the mobile phase, the components of that solution migrate according to the non-covalent interaction of the stationary phase with that of the column. Coming to the pumps. The role of pumps is to force a liquid called as mobile phase through the liquid chromatogram at a specific flow rate expressed in milliliters per minute. Normal flow rates is in HPLC are in 1 to 2 ml per minute in range. Typical pumps can reach a pressure up to 6000 to 9000 psi or 400 to 600 bar pressure. During the chromatographic experiment, a pump can deliver a constant mobile phase composition, that is either isocratic, or an increasing mobile phase composition, that is gradient. The types of HPLC. There are several types of HPLC pumps used for HPLC analysis. Most commonly are reciprocating piston pumps, syringe pump, and constant pressure pump. Reciprocating piston pump. It consists of small motor driven where the piston which moves rapidly back and forth in an hydraulic chamber that can vary from 35 to 400 microliter in volume. On the back stroke, the separation column valve is closed and the piston pulls in the solvent from the mobile phase reservoir. On the forward stroke, the pump pushes the solvent out of the reservoir. A wide range of flow rates can be attained by altering the piston stroke volume during each cycle or by altering the stroke frequency. So, this is the load position and injection position of the injector port. So, when you inject the sample through the syringe, it will enter into the loop during load position. Whereas in inject position, the sample whatever is present in the loop is sent to the column which can go for further detection. Coming to the columns. Column is considered as the heart of the chromatogram. The column stationary phase separates the sample components of interest using various physical and chemical parameters. It is usually made of stainless steel to withstand high pressure caused by the pump to move through the mobile phase. It is usually made of stainless steel to withstand high pressure caused by the pump to move the mobile phase through the column packing. Other materials include PEEK and glass. The small particles inside the column are called packing, which cause the high back pressure to the normal flow rates. Column packing is usually made by silica gel because of its particle shape, surface properties, and pore structure, which gives us good separation. God column. It is used to remove the particular matter and contamination. It protects the analytical column and contains similar packaging material. Its temperature is controlled less than 150 degrees centigrade and column length can vary from 5 centimeter to 30 centimeter in length. Coming to the detector. The detector can detect the individual molecules that elute from column and convert that data into an electrical signal. A detector serves to measure the amount of those molecules. The detector provides an output to an recorder or a computer that results in liquid chromatogram. Detector is selected based on light or on sample under detection. Commonly used detectors in HPLC include UV, that is ultraviolet. This type of detector responds to substance that absorb light. The UV detector is mainly used to separate and identify principal active components of the mixture. UV detectors are the most versatile, having the best sensitivity and linearity. UV detectors can be used for testing substances that are low in chromophores, colorless or virtually colorless, as they cannot absorb light at a lower range. They are cost effective and popular and widely used in industry. Coming to fluorescence detectors. This is a specific detector that senses only those substances that emit light. This detector is popular for trace analysis in environmental sciences. As it is very sensitive, its response is only linear over a relatively limited concentration range. As there are not many elements that fluorescence, samples must be synthesized to make them detectable. Next detector is mass spectrometry. The mass spectrometry detector coupled with HPLC is called as HPLC-MS. HPLC-MS is the most powerful detector widely used in pharmaceutical laboratories and research and development. The principal benefit of HPLC-MS is that it is capable of analyzing and providing molecular identity with wide range of components. Detector uses a monochromator and is one of the best sensitive LC detectors. This detector is extremely useful for detecting those compounds that are non-ionic and do not absorb UV light and do not fluoresce. Examples include sugar, alcohol, fatty acid, and polymers. Next is data processing unit or computer, frequently called as data system. The computer not only controls all the modules of HPLC instrument, but it takes the signal from detector and uses it to determine the time of evaluation, that is retention time of the sample components, that is called as qualitative analysis, and it also can detect the amount of sample, that is quantitative analysis. The concentration of each detected component is calculated from the area or height of the corresponding peak and reported. Retention time. In a chromatogram, different peaks corresponds to different components of separated mixture. The time elapsed between the sample introduction and maximum of response, it is the characteristic time it takes for a particular analyte to pass through the system. So the time taken for the analyte to travel from column inlet to the point of detection or peak maximum is called as retention time. Applications of HPLC. HPLC is one of the most widely applied analytical separation technique. It has pharmaceutical applications like during tablet dissolution of pharmaceutical dosage forms and for detection of shelf-life determinations of pharmaceutical products, identification of counterfeit drug products, and pharmaceutically quality control department uses mostly HPLC for its detections. Advantages of HPLC. Separation is fast and effective and it has high resolution power when compared to other analytical techniques. Continuous monitoring of column effect is seen. It can be applied to the separation and analysis of very complex mixtures. Accurate quantitative measurements can be done. Repetitive and reproducible analysis using the same column is also seen. Adsorption, partition, ion exchange, and exclusion column separations are excellently made. HPLC is most versatile than glass liquid chromatography in some respects because it has the advantage of not being restricted to volatile and thermally stable solute, and the choice of mobile and stationary phase is much wider in HPLC than GC. Both aqueous and non-aqueous samples can be analyzed with little or no sample pre-treatment. A variety of solvents and column packing are available, providing a high degree of selectivity for specific analysis. It provides a means for detection of multiple components in single analysis. These are my references for doing this PPT. Thank you one and all for the patient listening.